Hello, everyone, and welcome to this webinar on Power for Complex Hypotheses, which is the latest in our monthly webinar series. In this webinar, we will cover complex hypotheses, by which I mean non-inferiority, superiority by a margin, and equivalence hypothesis tests. So we are going to discuss design considerations for complex hypotheses, including use cases, choice of margin, and sample size determination for each of the three hypothesis types. We also take a look at a couple of practical examples of sample size determination for a selection of endpoints for the different hypothesis tests in nQuery as well. But before we get to that, I'm just going to run through some quick housekeeping bits first. So today's webinar is being recorded, and the recording, along with the slides used, example files, etc., will be sent out to you shortly after we conclude here today. If you have any questions, please feel free to type those into the chat box, and I will do my best to leave room to answer those for you at the end. However, if we do run into time constraints, I will be sure to answer any questions that I don't get to via email as soon as possible. And the same will go for any questions that you might send after the webinar is finished. I will respond to those via email as well. You may also use the chat box to inform us if you're experiencing any technical difficulties, and one of my colleagues will do their best to resolve them for you as quickly as possible. So before we go any further, just by way of a quick introduction, my name is Dennis Desmond. I'm a research statistician here at StatSolve, and I will be your host for this webinar that is brought to you by nQuery. Also, I'd just like to start by saying thank you for taking the time to join us here today. So just give a very quick rundown of the agenda for this webinar. Firstly, we'll start off with a quick introduction to the topic today, complex hypotheses. And I'm sure most of you are familiar with each of these hypothesis tests, but we'll be covering it just to make sure we're all on the same page. I'll give a brief overview of each and review some terminology as well. And then we'll go into some of the considerations for non-inferiority and superiority by a margin testing, including some worked examples, before we then move on to equivalence testing. And again, there'll be a worked example here too. Finally, we'll wrap things up with some discussion and conclusions, and of course, some questions as time allows. So, as I mentioned, this webinar is brought to you by nQuery, the complete solution for optimizing your clinical trial designs. nQuery is very widely used in pharmaceutical and biotechnology industries and offers sample size determination power calculations from preclinical research through to post-marketing studies, with the main focus being on confirmatory phase 3 trials, and features over 1,000 scenarios for fixed-term, Bayesian, and adaptive studies. If you're interested in learning more, you can see the resources section on our website for demos, work examples, previous webinars, and of course, future webinars as well. So nQuery operates on a tiered licensing model. So each of the 4 tiers gives you access to a different feature set depending on the tier which you purchase. Just note that this is a progressive model. So if you buy or upgrade to a higher tier, you will have access to all the features in the lower tiers as well. And so the basic tier is available to all customers who purchase an nQuery license. This will focus on sample size and power calculations for a variety of designs, endpoints, and hypotheses. The Plus tier focuses on our Bayesian features, such as credible intervals and assurance. The Pro tier, this is by far our most popular tier, and that includes the adaptive design features, such as group sequential designs, multi-arm, multi-stage designs. And then lastly, there's the Expert tier, which currently focuses on simulation tools for milestone predictions for enrollment and event milestones. If you'd like any more detail on this, you can follow the URL here at the bottom of the slides once they'll be sent out to you. And that will take you to our comparison tool page, which also includes a detailed breakdown of the tier required for each table offered in nQuery as well. On that note, if you don't already have a license or you would like to try a higher tier than you currently have, you can get a free trial of the full features of nQuery from our website. But I'll give you a bit more detail on that at the end, including some contact details in case you'd like to get in touch with us and get any further information from us. We'd be happy to help. So With that, I think we've all the housekeeping and introductory items covered. So we're going to start with the content of today's webinar. And as I mentioned, we're going to firstly start with a quick introduction and a brief overview of complex hypotheses, just to set the scene for the webinar today. So in clinical trials, the objective is usually to evaluate efficacy and safety in a new treatment. And a hypothesis is formed based on these primary study objectives. A hypothesis is a statement about the effectiveness or the safety of this new treatment in the population of interest, which defines which of the study variables are considered the primary clinical endpoint. And then that hypothesis can be tested using an appropriate statistical method. So, for example, in the typical inequality testing scenario, an example of a hypothesis is that there is a treatment effect. So in the means case, that the mean response in the treatment group does not equal the mean response in the placebo group. However, the definition of efficacy will depend on the clinical and regulatory scenario. So for another example, new treatment approval versus approval for a generic version of a pre-existing treatment. So it can be of interest to test if a treatment is non-inferior to, superior to, or equivalent to a standard treatment. And these complex hypotheses are what we're going to be looking at in our webinar today. And so another thing to clear up here is just some of the terminology. So in the literature, you may see inequality testing referred to as equality testing or superiority testing. And you can see there that could be a potential point of confusion because superiority by a margin testing is also called super superiority or superiority testing in some places. And so when you're testing for inequality, this is when the margin delta is equal to 0. And that's what's known as statistical superiority. On the other hand, in the superiority by a margin test, you'll have a value of delta for the margin greater than 0, and then this will be clinical superiority. So that's just something to be aware of. But just for the avoidance of confusion, we will use inequality and superiority by a margin terminology today. Non-inferiority then is nice and straightforward, and equivalence is often referred to as bioequivalence, just because that's probably the most common use case of equivalence testing. Okay, so now we're going to give a brief overview of each of these hypothesis types that we're going to be talking about. And I suppose a point to note is that in each of these, we're going to be looking to test if a new treatment is similar to a pre-existing one in some way. So for non-inferiority, the goal is to show that the new treatment is no worse than control by some pre-specified margin. And control here is going to mostly stand in for a pre-existing treatment that you wish to compare against. And so in the case of in non-inferiority, it's not usually a problem if your new treatment is superior to control. And in fact, in some more modern study protocols, you can see a multiple hypothesis plan where you start out testing for non-inferiority, and if things look promising, then you can step up to test for superiority. Now that can't be done on the fly, of course. Everything must be appropriately pre-specified in order to control the type 1 error. But it's just something that is becoming more popular. Now, generally, when you are testing non-inferiority, you are testing a direct effect measure with a quote-unquote good direction. So mortality or blood pressure, in the case where a patient suffers from hypertension, then lowering the blood pressure is good. Increasing the blood pressure is bad. And so if the new treatment lowers blood pressure more than the existing one, then that will, of course, still be significant within reasonable bounds, of course. And so the main requirement that is an additional one to the inequality test is the addition of this non-inferiority margin, which is the effect size below which you would find a new treatment to be inferior to control. So in this figure on the right here, you can see that we have two scenarios A and B, and scenario A, you can see that non-inferiority is shown because the confidence interval is fully above this non-inferiority margin. However, in the second scenario, non-inferiority is not shown because the non-inferiority margin is within the confidence interval. And so then for equivalence testing, you are testing if the new treatment is equivalent to the control. And this is often done using an indirect measure. So by that I mean there's not necessarily a quote unquote good or bad direction. And this is where you get the concept of bioequivalence testing, because here you're testing pharmacokinetic parameters like the AUC or the Cmax, which are measuring the concentration of the drug in the blood over time. So the theory in this case being that if the generic drug has the same AUC profile as the off-patent brand name drug, then we can expect the same biologic effect. It's important though to note that you aren't verifying the treatment effect in a bioequivalence study, because it would take too long to redo a confirmatory trial to verify that effect. And so there will of course be various regulatory requirements for this type of testing. But in order to show equivalence, these pharmacokinetic measures cannot go too high or too low, because the original treatment was only shown to be safe and effective in certain concentrations, and the concentrations and the dose that it received approval for. So the goal is to have the confidence interval fall fully between the lower and upper equivalence margins. So again, you have a couple of scenarios here on the right, C through F, where you can see where equivalence is shown, and then here not shown in F as well. So then lastly, you've got superiority by a margin testing, and in a sense, a statistical sense, it's effectively the inverse of non-inferiority testing, because you're looking to show that your new treatment is better than control by a specified amount. But it is distinct then in terms of its clinical objectives. And so in an inequality test, you're looking to test a difference greater than 0, for example, but in a superiority by a margin test, you might be looking to test a difference greater than 0.2 or something like that. And a recent example that I think will be familiar to all is the COVID-19 vaccine trials. And in this case, the regulators required a minimum vaccine efficacy of 30% rather than 0. And so just a point to note, again, with the confusing terminology, this final scenario, G here, where it says superiority shown, this is an example where statistical superiority is being shown. If we wanted to show superiority by a margin, then the whole confidence interval would need to be above this superiority margin or super superiority margin to demonstrate the superiority by a margin in the new product. Okay, so now that we have given some context to the webinar, we can move on to part 2. And here we are going to talk about non-inferiority and superiority by a margin testing in more detail. And as I mentioned already, non-inferiority testing is a test to show that the new treatment is no worse than an existing one by a pre-specified margin. And we also saw from the figure on the previous slide that testing can often be framed in terms of confidence intervals being above a non-inferiority margin. But something I didn't mention was that the non-inferiority hypothesis is a one-sided test, which you can see here from the hypothesis statement. And so if you're going to use a test for the, or if you're going to test, for the non-inferiority at the 5% alpha level, then that will mean a 90% two-sided confidence interval. If you are going to use a one-sided confidence interval, then that will map 1-to-1. So that's just a small point to note. Non-inferiority hypotheses are very common for medical devices, generic drug development, and vaccines as well. And so, for example, if a treatment is less invasive than the current standard treatment, or perhaps significantly cheaper, or you might have an alternative vaccine formulation to increase patient options as well. So up to this point, we've spoken about testing against pre-specified margins, but we haven't mentioned yet how to select this margin. And this is a very important decision, and it is one that's subject of much discussion. And unfortunately, there is no golden rule that says do this and you'll be okay. And so there are various ways that you can select the margin, but essentially what you want to do is to make the choice based on a mix of domain expertise and then the prior data as well. So that would be earlier studies for this new treatment and also trials for the innovator treatment as well. So the ICH guidelines state that the margin chosen for a non-inferiority trial cannot be greater than the smallest effect size that the active drug would reliably expect to have compared with placebo in the setting of the planned trial. And so what the FDA suggests is using a fixed fraction M2 of the active control effect M1. And so that's the estimate of the standard treatment effect compared to control. Now, depending on the context of your non-inferiority trial, this M1 can take into account more than just this active control effect though. And so, for example, if the treatment is significantly safer or easier to administer, it may be possible, in discussion with regulators to justify a more relaxed choice of M1. By default though, conservative margins are going to be encouraged, and especially in the case of generic medicines, because you want your new treatment to do the same thing as the standard, and in most cases, when you've got confidence in the product, that'll be okay to select that conservative margin. Now, the final point here is that there's the strong assumption in a two-arm non-inferiority trial, so that's a new treatment versus standard, that the effect size from the existing treatment is the same as it was from its approval. But of course, treatment effect can vary in population for a number of reasons, such as age or region, etc. So there's a requirement to have appropriate trial conduct in order to ensure assay sensitivity. And so this requirement means regulators will look for the trial to replicate the conditions that were used in the standard treatment's approval. And so this would mean, inclusion, exclusion criteria, the logistics in terms of administration, scheduling, etc., and the region, things like that as well. And And so they will also look for some additional data in this study in order to justify your selection of M1. And in certain cases, they're actually going to look for a 3-arm trial, which is seen as kind of the gold standard in terms of assuring assay sensitivity in a non-inferiority trial. And so what this means is that you're going to have the addition of a placebo arm so that you can now have a direct assessment of the effect of the standard treatment versus placebo, and your margin can be a fraction of that. Now, because the addition of a placebo group to this design, it's only feasible to use a 3-arm trial when it's ethical to do so, because of the fact that when you're doing a non-inferiority trial, it means that there's already an existing approved treatment for whatever condition that you are looking to introduce your new treatment for. And so taking a patient off of this standard treatment to go on the placebo arm is only going to be an option in certain cases. Perhaps where the condition isn't so severe, that it's not really a problem to ask patients to enroll in a trial where there's a placebo arm, or perhaps in certain conditions the treatment might be so toxic that patients might have opted not to receive it. So obviously it's just going to be discussions with the regulator, and it's going to be dependent on your trial conditions. But for that reason, it's not a one-size-fits-all solution. Okay. So again, now we're going to look at superiority by a margin testing. And as I mentioned previously, it's a test to show the treatment is better than the existing one by some pre-specified margin. And again, same as before, it can be framed in the context of having the entire confidence interval being above the super superiority margin. And again, it's a one-sided test, and as I said in the overview, it's essentially the statistical inverse of non-inferiority testing. And so you could create a general version of a null hypothesis that would be delta less than or equal to the margin delta 0, and if delta 0 is negative, then it's a non-inferiority test, and otherwise it's a superiority by a margin test. But obviously the clinical conditions are different, and that in non-inferiority you're testing that your new treatment has some benefit for patients, even if it might be slightly less effective than the current treatment. Here in the superiority by a margin study, you need to show that your treatment is some amount better than the current, because it might be more expensive, or perhaps the standard treatment is administered orally, but your new formulation is administered via injection. And so to justify this in increased demand for patients, they can't self-medicate anymore, then there would need to be more than just non-inferiority or equivalence in this case. And so this is very common in vaccine studies, medical devices, surgeries, etc. And as I mentioned earlier, the example for the COVID-19 vaccines, it was an FDA mandated requirement to have a vaccine efficacy of greater than 30%, as opposed to a standard inequality test. Given the fact that you're looking to vaccinate the whole population, there's the cost of doing so, the logistics of the rollout, So, in this case, it's understandable that they're going to want, something stronger than just an inequality test for this setting. And again, like non-inferiority testing, the choice of margin isn't outlined explicitly in the guidance. Again, you're going to be basing it on expertise and prior data and discussions with the regulator. And this is all done to kind of determine what would be clinically meaningful for this margin. Now, it'll also depend on the disease or the condition in question as well. In certain chronic conditions, a potential increase in toxicity may be acceptable for smaller increases in efficacy than it would in a less severe condition. And so, on the whole, its patient benefit is the priority. And so, given that this new treatment has some drawback, the margin is going to need to have strong justification. And so, in terms of the other practical considerations, like ensuring assay sensitivity and the additional evidence in superiority by a margin and non-inferiority testing is very similar. And then they're also very similar in terms of, sample size determination, which we can take a quick look at here. And so the first thing to mention, before any calculation can be done, with these tests, you do need to explicitly specify your null hypothesis. And that's because, you're going to need to have major choice of margin in order to get the sample size. And so, in an inequality test, where it's, you're typically testing the null that the mean difference is 0 or the ratio is 1. Here, the definition of the null hypothesis, requires the choice of the margin, and so in order to do your sample size calculation, that choice needs to be made. And so then you're going to need to pick an appropriate design for your study, and for each of these endpoints here, this is just some of the most common ones. You're going to have a variety of methods available. And so obviously this table here is far from a comprehensive list. It's just not feasible to put everything on it, but it's a couple of examples along with a few references as well. And to start off, like in the means case, the analysis is actually quite straightforward, because due to the fact that the variance is independent of location, then in effect it's equivalent to a shifted one-sided inequality test. So if a standard inequality hypothesis for the mean difference is equal to 0, then here we're dealing with just a shift to the left or right, depending on the choice of delta. But the variance doesn't change depending on that choice. So from a power perspective, there's no problem. However, when you move into non-normal endpoints, things aren't going to be as easy. And that's mainly due to this variance location independence not being the case for these endpoints. So if you take portions, for example, the variance depends on the value of the proportion, so the shifting impacts the variance as well as the power and sample size calculations. And so in inequality testing, it's just the alternative that is shifted, and so the null case is this special case where odds ratio equals 1 or difference equals 0, and the derivations of the power formulae take advantage of that. But once you move to non-inferiority testing, then it does get much more difficult, because now the null is shifted as well, and this is going to impact the type 1 error. And so for non-inferiority and superiority by margin testing, as a result, there tends to be more options in terms of the methods for the non-normal endpoints, just because there will be different proposals for dealing with this variance location dependence. And, you know, in addition to all these new methods, you know, there'll also be extensions for common inequality methods, you know, such as chi-square for proportions or log rank for survival, that can be used as approximations, you know, if you're unsure about which method to use. So those might be more familiar, but, you know, they might not be as good as some of the methods explicitly designed for non-inferiority testing. And so, like I said, just a couple of quick examples here. You know, in the means case, you've got t-test, z-test, Mann-Whitney U, you know, fairly standard methods. And then for the proportions, Farrington-Manning likelihood ratio test is a very commonly used method. In fact, we're going to see an example using that in a second. You've got chi-square tests and exact tests as well. For survival, you've got log rank tests, Cox regression, linear rank tests like Fleming-Harrington. You've got max combo, and then the restricted mean survival time as well. And then for counts, you've got your Poisson negative binomial, but then you've also got methods like Anderson-Gill as well. So now that we've got that covered, we can actually move on to our first worked example of the day. And this comes from a study published in the New England Journal of Medicine, comparing two stents in diabetic patients. And so the authors say in this paper that drug-eluting stents markedly reduced the incidence of restenosis as compared with bare metal stents. And so this study was designed to show non-inferiority of the paclitaxel stent as compared with the sirolimus stent. Sorry for the pronunciations there. And so the primary endpoint was in-segment late luminal loss. And so on the left here, we have text from that paper outlining the sample size calculation. The margin, the non-inferiority margin, I should say, sorry, was set at late luminal loss of 0.16 millimeters. The expected difference was 0. The common standard deviation 0.45, and using a 0.05 one-sided significance level, they found that they would need 99 patients per group in order to demonstrate non-inferiority with a power of 80%. And on the right, then you can just see that information summarized in the table. And with that, excuse me, sorry, we are ready to switch over to nQuery. And we can open up the non-inferiority t-test for two means. And so just very, very quickly, for those of you that may not be as familiar with nQuery, this is how most of the tables in the software will look. On the left-hand side here, you have a row name, and they'll tell you what input is required. And each of these individual columns will act as a separate calculation. The yellow rows indicate which rows can be solved for. And you can also choose from the drop down here which you'd like to solve for, and it'll indicate which values you need to specify in order to get a result for that. Also, you can just fill in the calculations as you go, and once an appropriate, or the, excuse me, fill in the cells as you go, and once an appropriate number of cells have been filled in, the solver will auto-calculate for you. On the right-hand side here, then you've got some help cards for each of the different rows, and there's a home card as well, just to go through some suggestions and references in order to provide some further information for each of these inputs. So those are some of the basics, but the software is pretty intuitive, and you should be able to follow along with the rest of this demonstration fairly easily. So what I'll do is I'll just get started straight away, and we can fill in our one-sided alpha of 0.025, and then we have our margin of minus 0.16. I'll just move my mouse here, and then we've got the expected difference. Sorry, just go back and check there. It was 0. Sorry, and then we have the little convenient auto calculation here pop up for the difference in the differences. The common standard deviation was 0.45, and then we get another auto calculation here that brings up the standardized effect size. And then we were looking for a power of 80%, and you can see that sorry, significance level was 0.05, my apologies. You can see that we get a sample size per group of 99. So apologies again there, just for filling that out wrong, but as you can see, it's a fairly simple example, but it's a nice introduction, and we can go back to the slides now, and we can pull up our second example. And this example comes from the New England Journal of Medicine, and it is a comparison of ketamine and electroconvulsive therapy for patients with non-psychotic treatment-resistant major depression. And so in the paper, they explain that ECT has been one of the most effective strategies for treatment-resistant depression for almost 80 years, but it's underused due to its limited availability and social stigmas, etc. And so ketamine is an alternative to this treatment for patients, and it's one that is of interest to patients because it doesn't require general anesthetic. And so for this trial, the non-inferiority margin was set at 10%, with the total sample size of 346 patients, and they would achieve 80% power to detect the non-inferiority given a one-sided significance level of 0.025, 50% response rate in the ECT group, and that the actual difference in response is 5%. And so they also used the Farrington-Manning likelihood score statistic, which, as I said, is a very common score statistic used in non-inferiority for proportions, just because it's been shown to have superior power and type 1 error control because of that variance location dependency. And so we can show a bit more on that in a second, but we'll also just note that they used the normal approximation version of the Farrington-Manning statistic as well. So now if we go back to nQuery, we can open up PT19, which is our non-inferiority test for the difference of two proportions. And we can start off by filling in our significance level, and again, one-sided. But for this test type here, if we click on this drop-down, you can see that there are a number of different choices available for this table. You've got Z tests, with and without continuity corrections. You've got T tests, and you've got 3 choices of likelihood score tests here. And so we're going to choose Farrington and Manning's test as they did. But there's a number of different options available in the software. Now, for the power calculation method, I mentioned that they used a normal approximation, but just quickly on binomial enumeration, this is an option for calculating the exact power by summing the probabilities over all possible outcomes. So, if you were to iterate through all possible excuse me, sorry, scenarios for responses in each group, so, say if you'd 100 people in your trial, you'd go through, 0 response in the standard group, 1 response in the treatment group, 1 response in the standard group, 0 response in the treatment group, 1 and 1, and through it. So every single possibility. And then you will calculate the probability for that scenario based on the responses, the estimated responses for each group. And, you can calculate the power of the test by summing up all of these values. And so, just given that, obviously, this can take a little bit longer to run than the normal approximation, and particularly for small difference in proportions. And, once the calculation gets sufficiently large, the normal approximation is going to be used anyway, just one for computing time, but then also, as the sample increases, that approximation is going to get better and better. But so yeah, again, we're going to use the normal approximation for now, but just to mention that. And so we had here the non-inferiority margin was minus 0.1, and the actual expected difference was 0.05. The response rate in the reference group was 0.5, and we can fill in our sample size ratio, which was 1, and our power of 80%, and this gives us the 173 subjects per group, as the paper mentioned. And obviously you can double that to get the 346. So we can go back to the slides again, and we can go on to our third example, which is our example for superiority by means. And this was taken from a study for an adjustable intragastric balloon for the treatment of obesity. And so in this particular paper, the authors outlined that standard treatments for obesity, such as behavioral changes, pharmacological therapies, and surgery, all have drawbacks. So there's potential for intragastric balloons to help address the gaps in obesity treatment, given that they're minimally invasive procedures. And so in the trial, the super superiority margin was set at 4.5%. The mean total body weight loss in the control group was 3.3%. The common standard deviation was 6.6%, and the mean in the treatment group was expected to be 10.34%. So with those, a sample size of 240 patients randomized 2-to-1 treatment to control would give 80% power to demonstrate super superiority or superiority by a margin at the one-sided significance level of 2.5%. And so just to note again, when we have our table over here, nQuery is going to look for the expected difference. So that's just the difference in the two expected means here. And so that's the figure we're going to keep in mind when we go back over to nQuery. And so what we're going to open up now is our non-inferiority test for two means, which is essentially the same table as we had before, but this time it has the addition of the sample size ratio parameter, so it will allow for unequal group sizes. But obviously this is a non-inferiority table, but a useful feature in nQuery is because the superiority by a margin testing is effectively the inverse of an inferiority testing, as we've been saying. All of the non-inferiority suites of tables can be used for superiority by a margin testing just by changing the sign. And other than that, then the rest is the same as before. So if we fill in the 0.025, now our non-inferiority margin, our super superiority margin, excuse me, sorry, was 0.045. The expected difference was 0.0704. And then again, we have this difference in deltas being auto-calculated for us. The common standard deviation was 0.066. Again, this will give us the effect size calculation. The power was 80%, and the excuse me, sorry, sample size ratio was 2:1. And so now what we'll see here is that we get 81 and 162, which is slightly different to the 240 that they had in the paper. So what I'll do is, if I just copy these values over here, but instead I pop in exactly what they had in the paper, you can see that it's 79.93, which isn't a major difference. And this could be just rounding in the presentation of the means, with the percentages, or perhaps they just rounded the sample size down to the nearest 10, something like that. Not a big deal at all. So with that, we can go back over to the slides, and we're ready to go on to our next section, which is equivalence testing. And so just, given the fact that today we're looking at all of these complex hypothesis tests, this is going to be a more high-level overview of equivalence testing, much like it was with non-inferiority testing and superiority margin testing. But one thing I just want to highlight is that bioequivalence is going to be the subject of a future webinar. So do stay tuned for that, and there's going to be an opportunity to go into much more detail then. And so, as we've said already, the goal of equivalence testing is to show that a new treatment is equivalent to a pre-existing one. And the most common method for doing this is the two one-sided tests method, which you'll often see abbreviated as TOST. The equivalence hypothesis statement is such that we'll fail to reject the null if the difference in treatment is less than the lower equivalence margin or greater than the upper equivalence margin. And then we could accept the alternative hypothesis of equivalence if the difference lies between these bounds. So this is an interesting situation because in this case, the rejection region is bounded, whereas usually it's the acceptance region that's bounded. And so this is an important part of bioequivalence because of the fact that you aren't testing for a direct effect anymore. In the case of generic drug development, where bioequivalence is kind of the standard approach, and you can assume that higher concentrations are necessarily going to be better, like you can for a direct effect, with mortality or something like that. And so, as we mentioned, higher concentrations could lead to an increase in toxicity events or a decrease in efficacy. And so it's very important to have a similar concentration of the active agent in the blood, compared to the innovator drug during its confirmatory trial. And so another point just with the TOST method is that you're going to test both null hypothesis hypotheses at the one-sided significance level. So firstly, you'll test if it's less than the lower equivalence margin, and then you can test if it's above the upper equivalence margin or vice versa. But you do need both of these to be significant in order to find for equivalence. But the overall type 1 error is equal to the one-sided alpha for each test, without the need for any multiplicity adjustments. But if you want to use a confidence interval approach, like we saw in the figure at the slide at the beginning, then you'll need to use twice the TOST alpha because it's not a two-sided test. So for example, 5% TOST alpha would equate to a 90% confidence interval. And so, as I've mentioned a few times now, equivalence testing is most often applied in bioequivalence trials, where crossover trials, are most often seen. And a crossover trial is one where individuals receive more than one treatment over the course of the study. And so in the classic 2x2 case, there's two treatments, 2 periods, and the patient would either go A, B, or B, A with a washout period in between. There are also higher order equivalents, higher order crossover designs that allow for more treatments and periods and different sequences, but these are typically used for higher variable drugs, for example. Now, biosimilar trials are an example where kind of a standard parallel trial might be used. And so per the EMA, biosimilars, aren't the same as generic medicines. A biosimilar is a version of an off-patent biological medicine. And so given the built-in variability in creating biological medicines, just in the manufacturing and things like that, it's not the exact same as just creating a chemical replica as it is in the case of generics. And so there's more evidence that needs to be collected in order to show that two biological medicines are biosimilar. Another consideration for equivalence testing is the definition of equivalence to use. And so in most cases, this is going to be average bioequivalence. This is what is used in obtaining regulatory approval for generic medicines and things like that. And then individual and population bioequivalence deal with switchability and prescribability. Then there are different measures to consider, such as the area under the concentration time curve, for the active component in the drug in the bloodstream. You've got the maximum concentration Cmax and the time to maximum concentration Tmax. So in the case of bioequivalence, actually the margins are typically set by the regulator. So for example, in the case of the FDA, they often require the geometric mean ratio of the AUC or the Cmax to be bounded by 0.8 and 1.25. So this is because when you're dealing with ratios, you want the bounds to be reciprocal of each other. And so that would be typical, but of course, it can also be based on expertise or variance as well. It might be the case where the margin is scaled up for the highly variable drugs, which are drugs that have within subject variability of greater than 30%. And so I'm just conscious now that I'm running a little tight on time. So I'm actually going to, quickly move on to our last worked example, but I will just highlight here that we have a similar table for equivalence like the one we saw earlier. Again, with the references and various methods. And so sorry about that, but again, like I said, I am just conscious of time. And so this is our final example for today. And so this comes from a trial from a proposed denosumab biosimilar, MW032, and it's for the treatment of solid tumor-related bone metastasis. This term here, the UNTX over UCR, per the protocol, this is an abbreviation for a bone turnover marker that was the primary endpoint for the study. Okay, so the significance level that was used was 0.05. The expected mean difference was 0, and the equivalence margin was 0.135. The common standard deviation was 0.58, and with 634 total patients randomized 1-to-1, the study would have 80% power to test for equivalence. So we're going to open nQuery for one final time, and we are going to open up our two one-sided equivalence tests for the two-group design. And so we can start off filling in 0.05. Then we have our equivalence limit. Excuse me, sorry, I'll move the mouse again. We had then our expected difference of 0. We had the common standard deviation of 0.58, and then we fill in the power. And we give a sample size per group of 317 for a total sample size of 634, as was in the paper, sorry. And so, sorry about that, but I kind of flew through the example and through the sample size slide as well. But again, I'll just mention that we're going to have the whole webinar on equivalence and bioequivalence coming up soon. So keep an eye out for that. But we'll go back to the slides now, and we can start wrapping up. So we wanted at the start of this webinar, or excuse me, sorry, at the start of this webinar, we started out by discussing how the clinical trial landscape meant that inequality testing was the standard, but there was an interest in being able to test if treatments were non-inferior to, superior by a margin to, or equivalent to existing treatments, and this is where our complex hypotheses came into play. We also discussed the distinction between the clinical and statistical superiority, to clear up some of the common terminology used in the literature. And then we went into more detail on each of the individual hypotheses themselves. So in the case of non-inferiority, you're testing a direct effect with a good direction, like mortality. However, for equivalence, you're often testing indirect measures like pharmacokinetic responses that don't have that obvious good or bad direction. And then in terms of statistical measures, non-inferiority and superiority by a margin are very similar, but the difference is in their clinical objectives. And so margin selection was the kind of common thread between all of these hypothesis types. And, it's a very important aspect of study design. And, it'll often be based on expertise and prior data, especially in the case of non-inferiority and superiority by a margin testing. In non-inferiority testing, it's set as some fraction of the standard treatment effect, but it can also take account for the safety profile or secondary endpoints, and regulator advice as well. And that's obviously especially relevant in the equivalent sense, where the margins are a bit more prescriptive from the regulators. Lastly, we saw that sample size calculations were possible for a number of endpoints for these hypothesis types, and that once you start looking at non-normal endpoints, things become a lot more complicated, just given the shifted null hypothesis and that variance location dependence. Okay, so I'm just after going over time there, so unfortunately I just won't get to the questions now, but I will make sure to get back to them via email as soon as possible. Again, I am very sorry for that. And just later today or tomorrow, I'll make sure to answer all those questions for you. As I mentioned at the beginning, if you don't already have a license for nQuery, but you'd like to try it out, or you'd like to try a set of features from a tier that you don't currently have, you can go to statsols.com/trial to get a free 14-day trial with no download or install required. It can be accessed right within your browser, and there are no credit card requirements. Just need to provide an email address. This trial is going to give you access to the latest version of the software, and as well, you'll get access to all the tiers, so base, plus, pro, and expert, and you'll be able to try out tables for fixed term, adaptive designs, and milestone prediction as well. Again, if you have any questions, you can email them to info@statsols.com, and then myself or one of my colleagues will get back to you, depending on who's best placed to be able to help you out. Also, feel free to reach out to us if you have any feedback. If there's an area maybe that you'd like more detail on, or perhaps you have a suggestion for a future webinar. Speaking of which, there's also more information, additional content, such as our library of past webinars on a wide range of topics, and they're all available on the website statsols.com. So at the end of the slides that you'll receive shortly, there'll be a list of references as well, and a reminder that the slides of the recording and the save files for the examples that I used in nQuery are going to be sent out to you in an email as well soon. So with that, that's going to be all for today. And so thank you all so much for your time and for your continued interest in these webinars. We do host one every month, so we do hope that you'll join us again soon, and we do really appreciate your continued interest in them. So with that, I'm going to finish up for now, but thanks very much and all the best.